Today, I'd like to tell you how we visualize copy number alterations in spatial transcriptomics data. And I hope you can appreciate in this initial slide, there is a gene expression matrix here. Each column corresponds to an individual spot from a spatial transcriptomics array, and each row corresponds to a gene. The values in the gene spot matrix are the gene expression values in that particular spot, where red means the gene is more highly expressed relative to other spots, and blue means the gene has less expression relative to other spots. And when you look at this, I mostly see noise and would say it's very difficult to see any patterns of copy number alterations. But I want to walk you through today how we are able to extract those signals from this noisy gene expression data. Now, first, I want to walk through or discuss how other people tackle this problem. And perhaps the most popular software is Infer CNV, which has been used on spatial transcriptomics data, but was actually designed on single-cell RNA sequencing data. Infer CNV calculates a moving average of gene expression across the genome. The idea is that when you look at one gene to the next, there's going to be fluctuations in gene expression. However, contiguous genes, if there were a gain, should by and large have higher expression, whereas if there were a deletion, one would expect that across a large enough window of genes that there would be less expression. And so in this example from Tarash et al., each row corresponds to an individual cell's RNA sequencing data. And you can see that when you look at the moving average of expression, there is what appears to be a deletion on chromosomal RM1P, a subtle gain on 1Q. You can compare this to the copy number information that is inferred from whole exome sequencing. So this is DNA sequencing, and we can consider this the gold standard. And by and large, these copy number inferences, at least for a subset of cells that are likely tumor cells, seem to agree with the copy number inferences from the DNA sequencing data. And so we know that this approach works, but we hypothesize that it can be improved upon. And towards that goal, we developed CNVkit-RNA. Now, CNVkit-RNA works by the same principles. However, it has a unique twist. And the idea is that not all genes are as informative as others in informing in allowing you to infer copy number information. So for instance, this is a gene called TATA binding protein. And when you look at its gene expression on the y-axis versus its copy number on the x-axis from Cancer Genome Atlas project data, you can see that the gene expression generally correlates with copy number. This R value of 0.7 strictly means that 70% of the variation in copy number can be explained by variation in gene expression. And so if I told you the gene expression of TATA binding protein, you would have a very good idea as to what its underlying copy number is. Whereas this counterexample, SOX17, it is a transcription factor. In general, the gene expression levels of transcription factors are not necessarily tethered to their copy number, but are regulated by complex upstream signals. And indeed, here you can see the R value is 0, meaning that if I told you the gene expression level of SOX17, you would be no closer to knowing the underlying copy number at that locus. And so what CNVkit-RNA does is it does calculate a moving average, technically a moving median of gene expression across the genome, but it weighs each gene proportional to how informative it is based on these correlations from an independent data set, which is the Cancer Genome Atlas project. So I want to walk you through each individual step. And we could start with the raw gene expression values that I showed you in the introductory slide. Now, we did one thing before creating this matrix, and that is we grouped some spots. When you look at spatial transcriptomics data, it is not uncommon for there to be wide ranges in the number of UMIs or reads per spot. And this is, for instance, in skin, typically associated with cellularity. So you might have tumor cells which have a large number of sequencing reads from the spot overlying them, whereas the stroma, mostly composed of fibroblasts, more paucicellular, might have tenfold less reads. Generally speaking, it is not easy when you develop these gene expression matrices if there's too much missing data. So for instance, a spot is mostly missing data, meaning that there are no UMIs corresponding to those genes. To analyze these side by side, And so we recommend, and we've provided in our GitHub repository, scripts that allow you to group spots together. So you can group, for instance, fibroblasts that are from the same gene expression cluster and are contiguous, and analyze a group of 10 fibroblasts side by side with one tumor spot. So once you've grouped spots so that each spot has roughly the same number of reads, you can generate one of these matrices. And I'm not going to show you the gold standard data, but we have reason to believe that there are gains on 1p, 3q, on and on and on, as well as deletions on 3p, 4q, 5p, and on and on and on. Here, each one of these columns are a spot, and the columns with bars above them correspond to tumor spots just based on their histology and their gene expression clusters. And then within each one of these gene expression clusters, we've rank ordered the spots by the number of reads. And so that's why it's generally noisier data on the left and less noisy data on the right. And so can we see any signals in the raw data? Not really. Maybe you can convince yourself, here's a group of tumor cells that, there's could be a deletion of 3p here and a gain of 3q here, but you have to know where to look. It's not easy to appreciate these signals. What happens when we apply CNVkit-RNA? Well, this immediately cleans up the data. You can see, for instance, this gain on 1p here, the loss on 3p, the gain on 3q, and I'll let you on your own time look at more of these spot chromosomal arm combinations, but immediately, just by calculating the weighted median across these chromosomal arms, we're beginning to see some copy number alterations that we know should exist. But we do apply a few additional filters. So in step 3, we remove spots with missing data. So even though we have pre-grouped spots so that they have similar numbers of reads, it is still possible to have a spot, for instance, with literally no genes that are expressed on an entire chromosomal arm. It's not many. If I toggle from here to here, you will not see huge differences in this matrix, but we do believe that filtering out spots that have too much missing data does improve the quality of your copy number inferences. And then lastly, we recommend that you center your data. And that's what we've done here. And I hope you can appreciate that some of these signals are popping out a little bit more. In the original gene expression matrix, each gene's expression was relative to all of the other spots in the matrix. So for instance, let's say you have a gain on chromosomal arm 3q. Those genes will appear to have somewhat higher expression, but at the expense of other genes, which are copy number neutral, they'll appear to have lower expression. And so the first pass of copy number analysis, that would show up as a subtle gain in some spots and a subtle loss in other spots. Now, if you believe you know which spots are tumor spots and which are not tumor spots, more importantly, which spots are not tumor spots, you can set that as 0, and that will take this subtle deletion and move it to and move all of the signals up so that it now appears as if it's copy number neutral, whereas the tumor spots will have a stronger signal. And then lastly, we have scripts that allow you to rank order the spots by their similarity to a gold standard, if that exists. And so here we did have whole exome sequencing of the DNA. And as I mentioned, we believe that there's a gain on 1p, loss of 3p, gain of 3q, on and on and on. And here we've identified the chromosomal arms with gains and those with losses. And so for each spot, we simply summed the amplitude of gains and we subtracted the amplitude of losses. And so if a spot has these gains and these losses, it will have a higher score and therefore appear higher in this rank ordered list. And so now my colleague Li-Ming Chen is going to walk you through the individual scripts that we use to carry out these analyses. Thank you.
Now I'm going to show you how to analyze copy number variations of spatial transcriptomic data. Before watching this video, I suggest you watching another video we made for point mutation analysis included in the same channel. In that video, we covered how to install the package stmut and showed you how to generate the gene expression profile for each spot, and also a bunch of CSV files. Some of them we will use in this video. Now let's move to copy number variations of bulk cells of the tumor sample. If you did the analysis using CNVkit, you will have one CNR file and one CNS file. In our case, we have both the CNR and the CNS files ready, we also provide an R function called bulk CNVs. This function gonna visualize the copy number variations of your bulk cells. And we can see there is 3P loss and a 3Q gain. Keep in mind, for now we have been talking about the copy number variation of bulk cells of the tumor sample. Next, I'm going to move to copy number variation analysis of spatial transcriptomic data. The first step is to group spots. This is an optional step. So why we do that? Because some spots have low coverage, which means those spots have low quality data to input copy number information. And there's pretty high chance those spots will be filtered out for downstream data analysis. To save those spots, we suggest grouping spots from the same gene expression cluster to get new spots. So we provide a function called spots grouped to group spots. This function takes 5 arguments. The first one is a data frame from a file called spot barcode summary.csv. If you watch the point mutation analysis video, you will know how to generate this file. The next 2 are output of a space ranger pipeline. One is filtered feature barcode CSV, which is the gene expression profile. The next is tissue positions list you can find in the spatial folder. The other 2 arguments, one is total reads, which defines the total reads of the new spot. The by default value is 5K. The other one, number of spots equal to 8, which means under the hood, this function will take spots from 8 rows away and 8 columns away. Then it will calculate the point distance from your current spots to the rest of the spots. Among those spots, then it will determine which spots are the closest, and it can be grouped into a new spot, which has a minimum read count of 5K. Once finished running this function, it will generate a folder called CNVs in a working directory. And then inside this folder, there should be another 2 folders called spot index, which is a list of new spot index. Another folder called txt, which lists the new spot gene expression profile. And also a list of other CSV files about this new spot. The step 2 is to infer copy number information over each spot by running the CNVkit. So we provide our example code here, import CNVkit import RNA. If you don't know how to run this CNVkit, we also provide a link to a video teaching you step by step how to run in CNVkit. Generally, CNVkit takes the gene expression profile. In our example, for this spot gene expression profile, it has 2 columns, a list of genes, and the read count for each gene. And the command take this gene expression profile to generate a CNR file for each spot. And the format of the CNR file, you will see the copy number information in the log2 column, and also another important column called weight, which defines the weight of each gene, which means how important it is in inferring the copy number information of each gene. This leads to our step 3, which we will generate the weighted arm based CNR file. We create a function called weighted arm media1 to do this. So what this function does is it incorporates the weight information into the copy number estimate by calculating the weighted median of copy number inference over each arm. And it will generate a weighted CNR file. Once you generate the weighted median based CNR file, then you will run CNVkit again to generate a CDT file. And this CDT file is the one of the final copy number information, which you can visualize in Java Trivial or in R. But we also recommend to run step 5. This helps you to better visualize the copy number information of your data. We create a function called CNV sorted. This function takes 6 arguments. The first argument is a data frame. And we provide our codes here for your reference to generate this data frame and make sure your input has the same format as ours. The second argument is a CDT file that you just generated. The third is also a data frame from a summary.txt file. This file is automatically generated when you run step 3, and it is saved in the same folder as the CNR file. The next 2 arguments on and the gain loss. These 2 are information of your bulk CNV information. So on is a factor list all the arms that have copy number variations. Gain and loss is a factor including either 1 or -1. So we observe 3p loss. So we assign -1 to it, and we observe 3q gain. So we assign number 1 to it. The last argument is tumor cluster. So you define which clusters are tumor clusters. So once you run this function, it will generate a new CDT file, which you can visualize in Java Trivial. And you will see, in our case, the blue color means copy number loss, and the red color means copy number gain. And you can also visualize this data in R, and we provide an example here, and you'll see the same pattern. For more details, please check our GitHub page. And next, I'm going to talk about the allelic imbalance. I'll see you in the next video.